Ladies and gentlemen, honorable laureates, dear colleagues, dear students, ladies and gentlemen, this year's Nobel Prize in Chemistry is all about outstanding imagination, creativity, and breaking new grounds, completely new grounds in science. Fundamental science. And as you know, the prize is awarded to three venerable gentlemen: Jean-Pierre Sauvage, Sir Fraser Stoddart, and Ben Feringa, for the design and synthesis of molecular machines, the world's tiniest, smallest machines you can ever imagine. So just think about that for a moment, about the machines. What is a machine? Well, you can define it as a type of construction composed of different components, different parts, working together and designed to produce some kind of useful output as a response of, well, typically energy input. All these parts, they have to be very, very carefully designed. They have to be built. They have to be assembled. They probably need to be able to move relative to each other while interacting with one another. This task is quite hard to achieve already at the macroscopic level. But just imagine what a very challenging task it is to do this at the molecular level. You know, you can only, in principle, only see these structures through very, very, very sophisticated microscopes. Nevertheless, our three laureates, they took on this, I would say, rather wild and formidable challenge of realizing these kind of machine-like structures at the molecular level. And this challenge not only required exceptional creativity in chemical synthesis and the design of chemical molecules. And all the laureates had to develop fundamental new methodology to achieve this. But it also required that they develop solutions for the controlled motion of these structures, these constructions, to result in machine-like function. So you will hear a lot more of these very spectacular structures and constructions that three laureates have made during these years very shortly. So they will tell us much more about their adventures and their journey towards functional molecular machines. And so I will now introduce our first speaker. And it's actually a little bit fitting that the name of our first speaker, Professor Sauvage, reminds us of the quite wild and difficult challenge this has been. Okay? Jean-Pierre Sauvage was born in 1944 in Paris, in France. He moved around quite a bit when he was young, but finally ended up in Strasbourg, where he was awarded a PhD in 1971 from the University of Strasbourg in northeastern France. And now he is a emeritus professor at the University of Strasbourg and also emeritus director of research at the National Center for Scientific Research, or CNRS, in France. So by that, I want to welcome Professor Sauvage up to the stage.
Thank you, Olaf. Well, let me start by saying that it's an incredible honor, to be here today and to deliver one of the three Nobel lectures in chemistry. I mean, this is something I never dreamt of in the past, I should say. And I would like to thank the Royal Swedish Academy of Sciences, for selecting our topic, molecular machines, and also for selecting me as one of the laureates with the two other laureates who are, in a way, good friends of mine. So let me start. I will also talk about chemical topology today, because chemical topology, it's really, to some extent, the precursor, at least for my group, the precursor to molecular machines, and which is, certainly something important. Just a bit of history. I think history of science is very important, and I deeply regret that very often it's neglected in our teaching. Chemical topology started many years ago in 1961 by a very nice publication co-authored by Dr. Frisch and Dr. Wasserman. And these two people, these two scientists, were working in industry. So in a way, it's kind of paradoxical that Bell Laboratories was financing research on something which looks completely exotic, in a way, and very far from applications. And Frisch and Wasserman just published a discussion. There were no experiments, nothing in there, just a discussion on chemistry and topology. And it was the first time that those two words, actually, chemical and topology, were associated. And thus, I think it is a very important paper, because for the first time, chemists could see that some molecular graphs, or some graphs, can be planar or non-planar, although the molecules could contain exactly the same sets of chemical bonds and atoms. So I think that was the very beginning. And now experimentally, I think the most important contribution comes from Germany, from Freiburg in Germany, very, very close to my own city, Strasbourg, maybe 80 kilometers southeast. And in particular, Professor Schill and his boss, Professor Lüttringhaus, a legendary organic chemist, Professor Lüttringhaus, they published a paper in 1964 in Angewandte Chemie. Peter, I hope you are sensitive to that. And it was a very important paper, historical paper. That was the synthesis of the first catenane, two catenane consisting of two interlocking rings. It was brilliant work, very, very elegant, but in terms of, let's say, practical outcome, it was not so spectacular because they needed many, many steps, and the overall yield of the process was certainly very modest. And so the people at the time were very impressed by the work, but they were not so enthusiastic trying to make catenanes themselves using strategies similar to those published in this historical paper. And for some years, maybe 10, 15, 20 years, the field became less active. And at the beginning of the 80s, when my group started in this field, it was really a dormant field. So nobody was working in this field. And Professor Hansson, in one of his letters, suggested that we may, or we are invited, in a way, to talk about the story behind the discovery. And I think it is a very good point. It's an important statement. How did you get the ideas? How did you start the work? And that's what I'm going to explain you. So at the beginning of the 80s, when I started my own group, I was very much interested in inorganic photochemistry. trying to split water, the water molecule, to H2 and O2. And of course, it was a big, big challenge, which is still a challenge today. And there were many important transition metal complexes which were expected to be important and interesting in relation to water splitting. And I had done some work in this field, in particular with my supervisor, Jean-Marie Lane. And the main complex was a ruthenium complex. And ruthenium is very expensive. And we thought that the copper complex could be practically more interesting. And if applications were expected, copper would be certainly more attractive than ruthenium. And Dave McMillan spent a year in Strasbourg on sabbatical leave from Purdue University. And we were making some strange molecules, strange to him, relatively simple to us. And when he saw this molecule, which we had made in my group, he said, we must collaborate. We are photophysicists and photochemists. You are molecule makers, in a way. And so let's collaborate. And we collaborated. We made a copper complex of this type containing 2 diphenyl phenanthralines. So this is phenanthralene. This is jargon. You can forget about it. But there are 2 nitrogen atoms here which are able to interact with copper. So we made this complex, and it turned out to be very exciting in terms of photochemical properties, in terms of photophysics. It had a long lifetime for its excited state. It could transfer electrons. It could do many, many very interesting things. So that was the very beginning, because if you look carefully at the structure, you realize that if you connect this point to that point, separately this point to that other point here, you make a catenane. Very, very simple. Let me do it with a better laser pointer. If you start from here, you will connect this point to that point, separately this point to that other point, and you have a catenane. And so we decided to, in a way, to stop, more or less, inorganic photochemistry, and we jumped towards organic synthesis and towards the making of catenanes. And I think the main requirement was to be experimentally able to jump from inorganic photochemistry to organic synthesis. And at the time, I had a very good friend, Christian Dietrich Büchecker, and this very good friend was a CNRS researcher in another group, and she decided to come and work with me. So basically, we started the group almost together, and she was a great organic chemist, a fantastic synthetic chemist, and thanks to her, all the work I will now discuss could be done. So we published the first paper in 1983. It's here, 1983, in a, let's say, reasonably good quality journal, but not a top journal, not a high impact journal. And we published it in French. That could be surprising. We published it in French because we had a feeling that it was perhaps it would become an important paper. I mean, it's pretty easy to understand what it means. You know, une nouvelle famille de molécules, les métallocaténanes. Do I need to translate? So that was the very first paper. And I will now explain you a little bit more in detail how we proceeded. So we, after this first paper, we really generalized the concept, which we call a transition metal templated principle. So we will use a transition metal as, let's say, the templating species able to gather various organic fragments to orient them in a 3-dimensional space and to maintain them in a given geometry. So it's quite easy here. We start from the transition metal center. We have two organic fragments able to interact with the metal. We generate what we used to call the entwined structure. We make a ring here, we make a ring here, and of course, at the end of the day, we obtain a catenane. Very, very simple. We could also be a little bit more sophisticated. We start from a ring which contains a coordinating fragment here. We mix with the transition metal, the open chain fragment, which is able to interact with the metal. And so we will obtain this assembly. And finally, we make a ring from here to here using a GG fragment. And we used to call that the gathering and threading strategy. If you like, I think nowadays we would say the assembling and threading strategy. Just a brief look at the molecules. So we start from the molecule, a very simple molecule, again containing this building block here, the phenanthroline with its two nitrogen atoms. Just in passing, this is not chemdraw, this is Indian ink drawing, a very old drawing, historical drawing, and in a way I think it can compete with chemdraw. So we take two such molecules, copper-1, we generate the entwined intermediate, as I said before. This compound can be made at the 10-gram scale within a couple of days. So this is very, very easy. We do not even isolate this species. We go on, and now we proceed to the double cyclization reaction. We take a long organic fragment. No need to discuss the methodology. We will interconnect those 2 points here and separately those 2 other points at the back. So this is exactly what we have seen on the strategy slide. And we can make, let's say, 0.5 gram of such a molecule, which is a catenane. Now we have indeed our 2 interlocking rings and a central copper-1 metal here. The yield was not very good, but I mean it was so straightforward that in a way it changed completely the situation, and I think it changed also the way chemists were looking at catenanes. So they were not completely exotic species. They were not completely impossible to make species. They became all of a sudden accessible molecules. We were lucky enough to crystallize those species. So this is the copper complex catenane. You see the X-ray structure, which is really kind of a picture of the molecule, a real picture of the molecule. I can move it so that maybe you can see it better. The copper-1 center is green here. In passing, this is ridiculous because all these complexes are deep red. But we had a green ball here to put. And if you demethylate, the system rearranges completely. And this is reversible. And once you have rearranged the system, you have a very open structure. The 2 rings can glide freely within the other. You have very little control over the geometry of this species, but it's a complete metamorphosis. And this is the way we call the process a metamorphosis. So this being said, I should say that we became interested in catenanes for several reasons, but probably the main reason was that catenanes are knots. So this is a knot, this is a catenane, a slightly more complex catenane than the one we have seen. They are ubiquitous in nature, in DNA, in proteins, in viruses even. We have no time to see so many examples, but here just DNA forms beautiful topologies and continuously. I mean, it's not something which is really exceptional, it is something which is routine. So a bit of history. So there were, of course, a lot of interest triggered by the synthesis of catenanes. I would like to refer to the beautiful work of our good friend Fraser, who really blew up the field in a way. So we started as a modest, small group in France, and I think basically there was an explosion thanks to him and his group. In the UK, Chris Hunter and Fritz Föckley, they use hydrogen bonding as assembling entities. Makoto Fujita did also beautiful work based on the palladium nitrogen bond, which is labile and thermodynamically stable. And I think I stopped in 1994. And after that, there were too many contributors, so there would be a too long list, you know. But there was beautiful work which is still done now. And I would like to thank all the people who joined the team, so to say, the worldwide team of catenanes and rotaxanes and other species. Just in passing, there was a pioneer, Hiroshi Ogino at Tohoku University in Sendai, who also published a beautiful piece of work with cyclodextrins and transition metals. Just before we start the molecular machinery part of my short lecture, a few topologies which have been made in my group, you know, mostly for the challenge that making these topologies represented. So that was the first system, a catenane, a 3-catenane, a very simple topology. In 86, 87, we had the X-ray structure in 87, and that was the cover page of Angewandte Chemie. The trefoil knot was very, very challenging. It took us years and years, but thanks to Christian Dietrich Büchecker, you know, we succeeded. And the doubly interleukin catenane, which is represented here, was mostly the work of Jean-François Nierengarten, another great synthetic chemist. Now let's talk about molecular machines shortly. And just referring to biology, because biology is really a source of inspiration for chemists. We knew that in biology, molecular machines are everywhere. Again, you cannot think of any important biological process not using motor proteins, so molecular machines. So ATP synthase is an example, and there was some beautiful work done in many places on ATP synthase and the understanding of its function and the way it works. Kinesin is also an important, a very important molecular machine with a small protein which can walk on a rail, the rail being a microtubule in the cell. So there are many, many, many examples. What about catenanes and rotaxanes? So catenanes and rotaxanes are certainly very attractive in this respect. You can quite easily figure out that a ring can glide along the axis on which it has been threaded from a position, let's say, to another position, and vice versa, go back to the original position. A ring could be hopefully able to rotate about the axis, or again, in a catenane, a ring perhaps can glide, can rotate within the other ring. So very attractive structures for making molecular machines. I know it's a very dangerous slide because maybe in the audience there are people who had done some beautiful work in the field of molecular machines, but I tried to collect, you know, some of the names of the most successful groups in the field of molecular machines based on interlocking, on catenanes and rotaxanes. And Fraser, Stoddart and his group, Vincenzo Balzani, Akira Harada, I mean, they have done beautiful work. David Lee, who was a student with Fraser, and many, many other groups nowadays using catenanes and rotaxanes to make molecular machines. So now I will just show you what we did in Strasbourg in relation to molecular machines. So the idea was quite simple. It was to take a catenane, a relatively simple catenane, and to move it. And the way we did was the following. So we have copper 1 here. And you know that copper has 2 oxidation states, copper 1 and copper 2. Copper 1 likes to be 4 coordinate. It likes to be surrounded by 4 nitrogen atoms so as for it to be tetrahedrally surrounded by nitrogen atoms. Copper 2 plus, on the other hand, is much more demanding. Copper 2 plus likes to be 5 coordinate or even 6 coordinate. 6 coordinate with an octahedral geometry, with Yan-Teller distortion, or 5 coordinate. And so now we will play with that. The requirements of copper 1 are not at all the same as those of copper 2. Let's start from copper 1. Here we have a very happy copper 1, and the copper 1 ion can be oxidized to copper 2 plus. But this copper 2 plus is still 4 coordinate, tetrahedral surrounding, and this is not satisfactory at all for copper 2. So the system will relax so as to find a more satisfactory situation in terms of thermodynamics, which means that the ring will glide here. The 3 nitrogen atoms which are here will replace the 2 nitrogen atoms which are here originally, and the ring will glide a half turn so as to generate a happy copper 2. So copper 2 is now 5 coordinate. It forms a very stable complex, and nothing moves anymore, apart from Brownian motion, of course. And this is very pleasant also to monitor for the people who did the work. We start from a deep red complex. We obtain a deep green complex. You have to be patient. It takes a minute or so. And after the deep green complex, you obtain a yellowish complex. Not a very nice color, by the way. And if you go back, you reduce, you inject an electron in the copper 2 center, you generate a 5 coordinate copper 1, and this 5 coordinate copper 1 is very unhappy, and the complex will rearrange so that the ring will glide again, and you regenerate the starting state of the molecule. So that was the first molecular machine in our group. It's not really a rotation, it's a pirouetting system. And I insist from the very beginning, we have no control over directionality. And if you want to know more about rotary motors, of course you have to read all the literature published by the group of Ben Feringa. I have a simple video, which I will show you. It's a video which was made by a student of mine who is now in the Netherlands. So we have copper 1. We have here a phenanthroline with its 2 nitrogen atoms. And here we have the second phenanthroline here. So this is copper 1 with 4 nitrogen atoms. Now we will abstract an electron. It's a blue electron, if you noticed. And the blue electron being removed, we have copper 2. The system rearranges, and you have now a 5 coordinate copper 2. You can re-inject the electron, not the same, of course. and you reform the 4-coordinate complex. And I think the beauty of the system is that you can do it as many times as you like with molecules. Of course, you can do it as many times as you like in silico. But with molecules too, everything is perfectly clean, perfectly quantitative, so that it's a nice, very reversible and clean system. So I will have very limited time to talk about molecular muscles, but let me just show you the principle. So we wanted to make a molecular system which is reminiscent of molecular muscles in the sense that contracting or elongating molecular muscles means gliding filaments. So you have filaments which glide along one another. So you have the relaxed form of the muscle here, the contracted form of the muscle, and you see that those filaments simply glide. And what we did was to design and make a molecule. It was a relatively complicated molecule. We made a molecule which behaved in exactly the same way. This is a rotaxane dimer, and what we could show is that we can contract it, and we send another signal to the molecule. We can elongate it and contract it, and this can be done, many times. We have no time, I assume, for 2 minutes. Thank you. It's very accurate. So we have no time for discussing, the work in detail, and I will skip all that. Some very important slide, I think, will be the slide gathering the names of the people who did the work. At first, I would like to conclude. This is my conclusion slide. Transition metals are very important. In our case, we are more coordination chemists than supramolecular chemists, to be honest. But transition metals played a very important role in our chemistry. They allowed us to build the system using templating principles. So the transition metal can gather and dispose the various organic fragments around the metal in a very well-defined and very well-controlled way, which allows to make target molecules of interest. And due to the presence of the transition metal in the molecule, the system has interesting electrochemical properties and photochemical properties. And this is how we could move the molecules, or let's say set some parts of the molecules in motion, such as 2 or 3 state swinging catenanes. We have seen the two state swinging catenane, molecular shuttles. We made a wing flapping rotaxane, and contractile and extensible molecular systems reminiscent of muscles. So I will stop at this stage. Let me just say one word of the French system. The French system, I'm not sure it's perfect, but there are good points in it, provided you adapt your policy to the French system. It can be efficient. And in particular, we have permanent people working in our groups, and we were working in my group as a group of permanent people. I was considered the leader of the group, but the other permanent people were friends, most of the time. Some of them were of my generation, and these are the people, working with me, not all of them at the same time, but Christian, Jean-Claude Chambron, who is now in Dijon, Jean-Paul Collin, who is now completely retired, Valérie Heitz, who is a professor in our university, and some younger members, CNRS members or assistant professors. So Jean-Marc Kern passed away in an aircraft accident, in a crash, many years ago, so that was also a very bad moment. And I have a special thought for my two first PhD students, Pascal Marneau and Romain Rupert, because I think at the beginning, it's important to work with very motivated students, and they were indeed very motivated. I'm not going to read the list of people who participated in this project or adventure on catenanes, rotaxanes, and knots. Perhaps the work on the muscles was mostly carried out by a Spanish postdoc, an incredibly active Spanish postdoc, Maria Consuelo Jimenez, and the work on the swinging catenane, the very first molecular machine, that was mostly the work of Aude Livoret. Finally, let me thank my university and the CNRS also, which had a very important contribution. The European communities, without the European communities, I don't think we would have been able to do as much work as we did. And the Région Alsace, which is around Strasbourg, my city. We have a foundation which was also very helpful. My new institute, because I had to move 5 years ago after I retired officially, ISIS, Northwestern University. Thank you, Fraser, for arranging for me to spend several years part-time in a fantastic environment. I would like to thank my supervisor when I was a postdoc in Oxford, Malcolm Green, a great organometallic chemist. The two most influential teachers, Guy Ourisson and Raymond Weiss. Finally, of course, my wife. She has always been very supportive. And this was also true for our son, Julien. And I would also like to thank the two other laureates, Fraser and Ben, because without their great contributions, I wouldn't be here today. Thank you.
Thank you so much. Thank you so much, Jean-Pierre, for that fantastic talk and an introduction to the world of mechanical bond, I would say. Right? Thank you. So it's time for me to introduce the second laureate, second speaker. And here is Sir Fraser Stoddart. And Sir Fraser was born in 1942 in Edinburgh, in Scotland, UK. He was awarded a PhD in 1966 from Edinburgh University. And he has also moved around quite a bit during his career, but he is now a Board of Trustees professor of chemistry at Northwestern University in Evanston, just outside Chicago in Illinois, USA. And just want to say one more thing, and I will welcome Sir Fraser up to the stage. That Sir Fraser is part of the so-called Fraser clan in Scotland, in north of Scotland, if I understand correctly. And the Fraser clan is well known for being extremely stubborn, but very trustworthy people. I think both of these characteristics are very good for science. But there is a third characteristic for the Fraser clan, and they are very, very fierce in battle. And I think maybe that is also something good for science, actually, sometimes. So by that, Sir Fraser, please welcome up to the stage.
Well, thank you, Olaf. Honorable hosts, fellow laureates, respected guests. It's an enormous honor for me to be standing here and sharing this platform today with Jean-Pierre Sauvage and Ben Feringa, the co-laureates that have been recognized by the Nobel Foundation and the Royal Swedish Academy of Sciences. I want to take you on a story that will reflect in some ways the citation, the design and synthesis of molecular machines based on the mechanical bond. Now, Jean-Pierre has already introduced you to catenanes and retaxanes. I like to use this word that was coined by First back in 1953, the mechanical bond, to describe a situation where within a molecule you have a physical bond between either two rings or between a dumbbell component and a ring. This is not a chemical bond, it's a physical bond, and as such, I think it's a very much revolutionary change in chemistry. We often refer to them as mechanically interlocked molecules, and I just put up this definition by Wikipedia to underline the fact that even Wikipedia appreciate that these are not supermolecules, they are molecules. And you see how they are derived from Latin, as is the two-ring situation. I want to segue into immediately, because we've had a marvelous introduction from Jean-Pierre, into a structure that we, when I say we, David Amabilino, who's in the audience, made in the mid-90s when I was at this marvelous University of Birmingham. And here you see a yellow component and a green component have the same chemical constitution, as do in a pair the blue and the red one. The black one is a slightly expanded version because it has to accommodate a yellow and a green ring here of the blue and the red. Here is a framework representation which shows the interactions that I'm going to be talking about primarily during my lecture between pi-electron-rich units, this dioxynaphthalene unit, for example, and these bipyridinium units. The molecular recognition that you see here is that you can find in DNA and in proteins. And in fact, these pi-pi interactions in this direction here, over here, and in this direction here, are aided and abetted in a huge amount by hydrogen bonds, and also, to a lesser extent, by CH-pi interactions, which you find in proteins. The olympiadine molecule, I should say, came to the world without too much effort on my part. As I say, David in the audience was the synthetic chemist, and the crystal structures you're looking at are the courtesy of David Williams, erstwhile Professor of Structural Chemistry at Imperial College London. You'll see that we went on and made a branched 7-catenane, and there is the crystal structure of that particular molecule. Now, at one time I was hoping that the Olympic movement would rally around and support my research, but the opposite was true. They don't like to see any use of their Olympic sign, as some of you may know from the response during the London Olympic Games to the butchers putting rings of sausages in their windows that were coloured like this. Okay, so I want to pay a tribute to Ed Wasserman, because it is debatable, maybe, I know that, whether this was the first catenane that was made back in 1960, but you can see as a result of an acylin, so-called condensation, and using some deuterium labeling, it could be argued, if you read this paper carefully, that this was maybe the first catenane to be made. But look, in a very, very small yield. This maybe is an overstatement of the yield in this reaction. Jean-Pierre already mentioned the very important publication in 1964 by Gottfried Schill from Freiburg University. This was a 22-step synthesis, of which you see in a telescope downway the last 4 steps of the synthesis. And this involved the use of classical chemical bonds, covalent bonds, to bring the pieces together, and then at the end of the day to cleave them. And one of my Bibles has been this book, catenanes, retaxanes, and knots, written by Gottfried Schill and translated, published in English in 1971. And now, of course, I pay homage to the remarkable breakthrough. It was a game changer, this French article in Tetrahedron Letters, which I did struggle to read a little bit. Where are you, Jean-Pierre? But we got the message from all these beautiful representations, and here I have perhaps put more detail in them. You've already heard this story from Jean-Pierre. So it was in 1989, after about a decade of developing molecular recognition around donors and acceptors, that we carried out a reaction in which we started off with this dication and a crown ether over here that I will call a template. And these two don't speak to each other. There's no molecular recognition. But as soon as this carbon-nitrogen bond is formed, you have a bipyridinium unit, and this wants to make its way inside this crown ether so that you can get donor-acceptor interactions augmented by hydrogen bonds. And then finally, another nitrogen-carbon bond formed, and a 2-catenate is the result. And the amazing thing for us in 1989 was that this arrived with a 70% yield. Now, the crystal structure, again, was carried out by David Williams, and I know that Peter Gillitz is in the audience. He helped to orient that molecule, I remember, in a pub in Sheffield, in a particular way that leaves it, I think, attractive. So now we go to retaxanes, and using the same type of thought process, here's our template now. We don't have a ring anymore, we have a dumbbell. And again, no recognition, but very easily, in a one-step reaction, you can take these chemicals here, and acetonitrile, and prepare, presumably in the first step, the same as the last time, with the catenane, and then see these non-covalent bonds coming into place in the retaxane environment, and again getting closure, and this time, of course, not with the same high yield as with the catenane, because this is a more flexible template, but still a very respectable 32% yield. Now, the importance of this discovery, and this takes us to 1991 now, is that this was the first example in our lab of what we call the molecular shuttle. This is a 2-retaxane where the ring can dart back and forth. You can see about 2,000 times a second at room temperature in acetone. The next challenge was to take away the degeneracy in that system and make it non-degenerate. In other words, turn it into a switch. So we replaced one of the hydroquinone rings by this biphenyl unit and by this benzidine unit. There's a story here. Birmingham was such a beautiful place, we were not allowed to carry out the chemistry using benzidine in Birmingham, and Richard Bissell had to go to University of Miami, where the restrictions on using benzidine weren't so high, and made this compound in Miami University under the guidance of Angel Kiefer. And now you can see we have a situation where the ring spends most of its time on this unit here, the benzidine unit, until it is protonated. And because the ring carries 4 positive charges, the ring moves away from the protonated benzidine unit to form another species where it is encircling the biphenyl unit. And all of this can be controlled, obviously, by pH. Now, I'm moving on to the first device that was made at UCLA in collaboration with Jim Heath. And what I want to show you here is, in a movie, how the synthesis is carried out. So this is now a tetrathiafulvalene unit, and this is a dioxynaphthalene unit. Here you see the templation of the blue ring around that unit. This is a hydrophilic stopper, and that's a hydrophobic one. And by, again, redox chemistry, we can put positive charge onto that TTF unit and have the ring move to the dioxynaphthalene unit. Equipped with this type of amphiphilic rotaxane now, we could fabricate a crossbar device in which, by hard lithography, you could prepare a chip with, say, 4 wires on it. This goes into a so-called Langmuir trough, and in the Langmuir trough, this monolayer of molecules is squeezed together until it's at a right level of pressure to when the chip is brought out by the so-called Langmuir-Blodgett technique, the monolayer is spread onto the surface of these 4 wires. And then more hard lithography is required, and all of this was done in Jim Heath's lab at UCLA and subsequently Caltech to give us a so-called 4x4 crossbar device. I go fast forward over a decade. We had a wonderful collaboration with Jim, and this is now a 400 times 400 crossbar device with wires around about 16 nanometers wide, and this gave us the basis for a 160 kilobit molecular random access memory. Jim was quite hard on the switches that he had. The threshold had to be at least 1.5 in this diagram here, so this meant that 3/4 of them, for one reason or another, were condemned, but at least a quarter remained, and that allowed him to punch out, using the ASCII code, California Institute of Technology, on a device where we have white blood cells here. Here is the device. Here are white blood cells. And so we're looking at a device that is, in its cross-section, smaller than the cross-section of a white blood cell. And this was 2007, roughly where the semiconductor industry is expected to be by 2020. One thing was not to our liking about these switches, and that is they don't last. They might go at best through 100 switching motions, and then the system just degrades. So I want to give you some very up-to-date results, and so this is a paper that's just been accepted for publication, and in fact has appeared now, in which we've used this bistable catenane with the same principle of playing off TTF against the dioxynaphthalene unit, placed inside a metal organic framework by a technique that has been featured and developed by my 2 colleagues, Joe Hupp and Omar Farah, called solvent-assisted ligand incorporation. So you just plug these switchable catenanes into the pores of what is called NU1000, this particular MOF. And you can see here, this is a film where you can see switching just by looking at the absorption around about 800 of the TTF unit, and that switching is taking place. I can't tell you how robust it is yet. The other development of our work at UCLA was in collaboration with Jeff Zink, where we put essentially rotaxanes on the surface of mesoporous silica nanoparticles and showed that we could use these, and I have a movie here to show you the incorporation of drugs under a concentration gradient, and then using the rings on the rotaxanes to close up onto the surface, hold the drugs inside, and then let them, in principle, be admitted into the environment of a cell. Here's the cell coming, and the capsule is inside, and when you give it a signal, by any means possible, then the drugs will spill out, and of course this is not going to be good news for the cell. Okay, so I'm going to make a fast transition now to molecular machines, and they are going to be based on this so-called flashing energy ratchet mechanism. And so this is where we need to have a situation where we can get unidirectional motion to begin with in a situation, and I'll use cartoons to begin with, where we use the cyclophane you're already familiar with, and we want to be able to bring it on to this dumbbell such that it comes over the neutral end, and after having done that, because of the recognition between the naphthalene and the ring system, to eject it by the right-hand side over the charged end. And this is achieved by a juggling around of energy barriers. So here is the lower energy barrier. It comes over the neutral one. And then by changing the oxidation state or reduction state of the ring and taking it to a bis radical cation, we reduce the positive charge and it comes off the right-hand side. So this is the way by which you can get one of the necessary things to be able to build a molecular machine, that is, getting unidirectional motion. Okay, so again, we are inspired by nature. I just want to point out that bacteria rhodopsin uses the same principle to pump protons across from outside to inside a cell. Here is the chemical constitution of the dumbbell that you've been looking at, and you've seen the cyclophane before. And again, this principle can be used to make what we've called a molecular pump. So this is the prototype that allows us to take the ring on from the right-hand side and leave it from the left-hand side. Anyway, we didn't have enough, as it were, driving force in this particular system, and we were able to change that situation for the better on the back of a discovery made at the end of 2009, published in 2010 in Stuart Cantrell's journal, and he's in the audience. And this amazing discovery that a tris radical trication can be a very stable species allowed us to be able to play off reducing against oxidizing conditions. And so one can contemplate now a situation where we turn the dumbbell through 180 degrees. The charge system on the dumbbell is on the left-hand side now and the neutral one on the right-hand side. And we're going through a sequence of reductions, relaxation, oxidation, relaxation. We could show that we can direct the ring from left to right. This form of templation can be illustrated by the making of the first homocatenane in the series of this so-called blue box, and this was carried out by Jonathan Barnes, who's in the audience, in a remarkable 41% yield based on the interaction that was such a surprise. It was so surprising that it's been described by one of my postdocs as an intellectually disturbing invention, as it were. And this monoradical can be further oxidized to a totally oxidized octacation. And there's the crystal structure of that one. How am I on time? 10 minutes, thank you. We can effect radical templation also in the rotaxane area by simply using a photosensitizer and a sacrificial electron donor, and the result is the formation again of this cis radical tricationic complex. And when you see these azide groups at the end, at least every chemist knows that you can carry out in principle a so-called click reaction with an appropriate alkyne, and we were able to do this, or Hao Li, now a professor in China, very efficiently. The point about this system is that when you bring it out of an inert atmosphere, this chemistry has to be done in, say, nitrogen or argon, then coulombic repulsion will take place and the ring will be pushed away from the formal bipyridinium dication. So here is the design of the molecular pump, where what you see going on here with this movie is the juggling about of transition states and energy wells to allow this ring to come on when it is in a reduced state, and then to be sent to the left-hand side over a barrier. We call this a speed bump sometime, and when we get it over there, it is such that it can't make its way back. And this is the, again, cartoon version, and we have a long collecting chain here. This is where we want to collect these rings and demonstrate that we're doing chemistry, as you'll see, away from equilibrium. In other words, we're moving away from molecular switches to molecular motors. Another movie here that shows the ring coming on from the left while it's being reduced, and then after being oxidized, going over the speed bump. And when the second one comes on, the first one wants to come back, but it can't because of the speed bump. So there's an enormous amount of delicate stereochemistry involved here and a huge amount of synthesis. The process that we're looking at, again, is one of consecutive reduction, oxidation, a little bit of thermal energy needed to push this ring over the speed bump into the collecting zone, and further reduction and oxidation and thermal process to bring the second ring on. So it's interesting that pumping one followed by two rings is possible. And in the context of doing this, we are more and more taking a system away from equilibrium. And what was very interesting was that we could carry out the first redox cycle here in a 90% yield. The second one, a little lower, 77% yield. Sorry, 85% yield. Overall, 77% yield for the production of this 2-catenane. And what I want to point out is the energy barrier that has to be crossed to put the second ring on is within experimental error of that for the first ring. And so what we're seeking to do now is to use this same type of approach, the radical chemistry, with a redox system that involves reduction, forming the very strong complex, and then oxidation, giving coulombic force that pushes it to the right. And you'll notice as this happens, I'm increasing the level of the energy here. And so we're going up in energy. We're doing work. And this is what the biological motors do as well. Yeah, that's the fourth one. So each cycle traps one ring in a high energy state. Some of the energy input is trapped, and we've gone from working with molecular switches to molecular spins because work has been done. Time 5, okay? Now, why would we be interested in going down this road? Because everybody's looking for applications. And let me give you a taste of what might be an application, so-called entropically dominated polyrotaxanes. And so now we're putting the pumping part of the molecule at each end of a long polymer chain. And using a light-powered system and even an electrically powered one, we can show, we're making progress here, that we can do work progressively and repetitively from each end of these polymer chains. And I take a movie here to show you what's going on. In solution, we have the redox chemistry, reduction followed by oxidation, forcing the rings on over this speed bump in two different parts, two ends of the chain here, so that we accumulate a lot of rings in an environment where they are both entropically and enthalpically unhappy. So what's the consequence of this? Well, before I come to that, I just want to summarize the principle that we're developing here, and that is to use a very stable interaction of a host-guest nature, and then after the reduction process to get this to happen, an oxidation process to switch on coulombic forces at a very big level so that we've got a highly unstable species, and this forces the units to come apart. So we have a modulation here involving kinetics of association and kinetics of dissociation. We're doing chemistry away from equilibrium. And the point I want to make here is that if you've been an undergraduate in chemistry or a graduate student in chemistry in the last 50, 60, 70 years, it's usually been all about equilibrium processes in the class. We've got to move now into the direction of studying systems that work away from equilibrium, because this is how the motors in our bodies work. So what are some of these big questions in this context? They are, what are the consequences of dissipation on materials properties? Well, we have a hint that these could be very useful, because in Japan, Professor Ito and Kato have, using cyclodextrins and polymers like PE ethylene glycol, polyethylene glycol, have been able to show that you can make these so-called slide ring materials. And these show properties of being scratch resistant. But we would like to be able to do this in a more widely defined arena involving organic solvents and organic type polymers. So we want to be able to design systems that operate repetitively and progressively away from equilibrium along the lines that I've just been describing. Okay, so just to summarize, this has been a journey that's taken at least 25 years, although I will just dip back for a little while at the very end to point out that to get to 1991 was already a decade of a lot of, shall we say, difficult experiments that seemed to take a long time, a lot of situations where things didn't work out the way we expected. But eventually, by the turn of that decade, when we went to Birmingham, we were able to make degenerate molecular shuttles and to put into them radically enhanced redox switches by 2010. We then could introduce unidirectional translation as a result of having these asymmetrical dumbbells. First of all, we used donor-acceptor interactions, but found that the discovery of the radical chemistry made it much more feasible to make linear motors, and we had to optimize Coulombic barriers after having optimized steric barriers there. The chemistry that was done here in making these energetically demanding transport systems ultimately led to the production of these artificial molecular pumps. And in this context, there are these early players, and I should recognize the very important contribution of theoretical chemist, computational chemist Bill Goddard at Caltech, because we do everything with a lot of computational work. Dina Stumian, who's been really our guide through all of this, physicist from the University of Maine, and then other people who, some of them in the audience, Jonathan Barnes, Christian Pizzato, Paul McGonigal, and Zhu-Yang Chen, who've really pushed forward our work on molecular machines. The leading molecular machinists, I would, of course, recognize right away my co-laureate, Ben Feringa, but I just want to put up with some trepidation, of course, the same that was expressed by Jean-Pierre Sauvage, other main players in this field, so Vincenzo Belzano, Alberto Credi, who's in the audience, my ex-student David Lee, Dina Stumian again, Joseph Miko, Colorado and Prague, Steve Loeb in Canada, Eddie Sivik in Australia at the ANU, McGill Garcia-Garabi at UCLA, Raphael Klein at the Weizmann Institute, Amr Flood in the audience, Ivan Ibrahimian in the audience, and I know that also Nicholas Giusepponi is in the audience. So I think the field is moving forward at great knots, and the recognition by the Nobel Foundation and the Royal Swedish Academy of Sciences will, I think, give it a great impetus. Can I just take 1 minute? Okay. Can we go back to Nobel laureate Alfred Werner? Because some of the introduction that I didn't put in because I thought I might not have time goes back to Alfred Werner and then a man called Joe Schatt, who was associated with Imperial Chemical Industries, where I spent 3 very productive years between 1978 and 1981. Before we go there, here is just a statement for the young people in the audience in a review by Chat that I think is worthy of some mention. Scientific discovery is not yet predictable. Note that. If you have an exciting road to follow, do not be put off by those who say there is nothing at the end of it. They do not know. Persevere and enjoy the excitement of exploring the unknown. And I think your 3 laureates in chemistry have all experienced some of the sentiments written there. In the audience is Howard Colquhoun from the University of Reading, who, when I first knew him, was at ICI And here is the crystallographer that I paid homage to earlier, and his student, Alex Lowen. And it was the entry into a field of second sphere coordination involving transition metals and bipyridine ligands that led in successive steps to organic systems that were very like the transition metal-based ones. And this happened in my lab during the 1980s. Finally, just to recognize the importance of the human and financial resource situation. First of all, this was my postdoctoral mentor in Canada, Ken Jones. This was the professor of the School of Carbohydrate Chemistry at Edinburgh, Sir Edmund Hurst, under whom I studied. He was a student of Walter Norman Haworth, 1937 Nobel laureate in chemistry, who was a student of Otto Wallach, 1910 Nobel laureate in chemistry. And they go back through Kekulé, and wait for this one in color. Do you recognize him? So I've got a good link to Sweden here. And back, of course, to Wohler, who challenged the vital force in his time. I just put up a few of the postdocs and graduate students that have played a big part in my research over almost 5 decades now, and also some of my collaborators at Northwestern, Berkeley, Usri Botros at Panacea Nano, who's sitting over there, and Roger Petman. Of course, it's not all done without some brass, as we call it in Britain. So I got a lot of support, ultimately, from this University of Birmingham. It was amazing when I went there in 1990, and also from Imperial Chemical Industries. Well supported by both universities in the United States, UCLA and Northwestern, and the funding agencies there. But last but not least, in recent times, the King Abdul City for Science and Technology in Riyadh in Saudi Arabia. And I will just close there and allow, if time permits, about 400 names to come and go in red and blue that have contributed to my research. I wouldn't be standing here if it wasn't for the contributions by this amazing group of people. You know, it's an enormous privilege to to practice one's hobby every day, along with young people, roughly between the ages of 22 and 32, and their names are going up there. I've been so privileged. I'm very privileged to be here. Thank you.
Thank you so much for this fantastic, inspiring, and animated talk, and also very visionary. And it's really nice to see some results that are really hot off the press. Right?
I always want to give results that are hot really off the press.
And also the link to Berzelius was fantastic. Thank you so much. So it's time to introduce the third speaker of this chemistry session, our third laureate. And here you see him. It's Bernard or Ben Feringa. And Ben was born in 1951 in Barger-Compascuum in the Netherlands. He received his PhD in 1978 from the University of Groningen in the north of the Netherlands. And then he kind of was lost for a while to fundamental science because he worked for a while in the shell industries. But luckily he came back, and he is now a professor of organic chemistry at the University of Groningen in the Netherlands. So please, Ben, welcome up to the stage.
Thank you very much. Members of the Academy, distinguished guests, ladies and gentlemen, it's a great, great honor to be here today. And I thank the members of the Nobel Committee, the Nobel Foundation, and of course the Royal Swedish Academy of Science for this honor given to me and to my colleagues. So, in the art of building small, which is the title of my talk, I will go in the coming half hour or so, walk from molecular switches to molecular motors. But before I do this, I would like to go back in time a little bit, widely over 100 years, because I often feel like the Wright brothers. And although people have been trying to fly for centuries, most of them didn't succeed until the Wright brothers were able to fly in this primitive machine for a couple of yards. And people were saying at that time, why should we fly? If God wanted us to fly, He would have given us wings. Nobody would have predicted that 100 years later we would build in Europe the Airbus or a Boeing 747 in the States. But remind you, although we get admired by the beauty of nature, the flying bird, a Boeing does not fly like a bird. I wouldn't be here in Stockholm. Neither would Fraser. The materials that is used to build this machine, the 3 million components, are completely artificial, synthetic, and the flying principle is completely different from this bird. But it flies perfectly, and it can carry 300-500 people across the ocean at high speed. But also we should feel very humble 100 years after the Wright brothers, because we still cannot build a flying pigeon. We cannot even build one cell of a pigeon. We cannot build even a component of the cell of the pigeon. So there is still some work to be done. I'm a synthetic chemist, just like my colleagues. We build molecules, and the power of synthetic chemistry is that you can build the materials that we use every day, our plastics, our fuels in our cars, the dyes, our drugs, etc. So being a synthetic chemist, we have endless opportunities to build, by this chemical synthesis, the new materials in our laboratories with specific properties, being it a drug, being it a fuel. And so we can design new molecules, and the opportunities are endless. And we engage in, for many years now, like my colleagues, in trying to control dynamic functions in molecules. And of course, we get inspired by Mother Nature. But let me go back to compare, when we talk about machines, a robot that we know from our car manufacturing plants, for instance, and the robot that is very active at this moment in your body, the ribosome. I would argue that this complex machinery in your body is way more complex than this robot that can precisely manufacture your car. But there is a distinct difference. This is hard material, this is soft material. And there is an effect of length scale. This is 2 meters and this is 24 nanometers in size, 1 billionth smaller. And this also, there is the effect that we have to work with molecules at low Reynolds numbers. The world is completely different. Here, we know we deal with gravity and weight, but at the molecular level, gravity doesn't play much of a role, and weight neither. It is the intermolecular interactions, interface phenomena, viscosity, etc. And of course, the Brownian motion, we should not forget. It's not so much about how to get motion, but how to control motion, because the Brownian motion brings us these Brownian storms of the molecules all the time. There's a lot of movement. So when we get inspiration from Mother Nature, as we did, we look at the machinery, and it was mentioned earlier, these beautiful motors that power our body, the cells, that make it possible, all these millions of motors that your muscles can move, that they work together, that the cells can divide, that things are transported in and out of the cell, that the ATPase can produce the fuel in your body, that the bacteria can swim with the bacteriflagella motor. And of course, maybe the most beautiful device, and that is the molecule that makes it possible that we can see each other. This is a molecular switch. This is a switch in your eye, and it's hit by light, and you can switch between two states. And this is where we started, and is where I started my journey in this field. So when you look at the eye, you have a 0-1 switch. You have a band molecule, and with light, it can switch to a more linear molecule that triggers a signal to your brain because this is embedded in a protein. But of course, it should be reversible. You should switch back and forth. Otherwise, we should see each other only once, and then it would be over. This process is extremely fast, and the fact that we can see each other is that millions of these molecules work together. So we built information storage systems many years ago, and the idea was to do computing or information storage. That was actually the basis idea. 0-1 switches using the principle that we use in our eye. Switching with light across a carbon-carbon double bond. Now you might wonder, why don't you use the molecules from your eye? But when you take them out and you try to make a device, they don't live for long. They are not stable enough. So we have to make more robust systems that you can switch back and forth. Then you might ask, how is it possible that you can switch along a double bond? Because we all learn in our first year undergraduate studies that a double bond doesn't rotate easily. But of course, with light, you can excite it, and very briefly you break the double bond, it rotates and it forms again. That is photoexcitation. That's exactly what happens in our eye. And we had the problem of non-destructive readout, because when we were switching with light, we were switching back and we were losing the information. And so what we did is we built these molecules left-handed and right-handed, like the two screws. Left-handed 0, right-handed is the 1. And we could switch between left and right. And that was the 0-1 principle that we designed. And that made it possible to do non-destructive readout. Then you can think of materials and what can you do with it. And we built actually plastics with these molecules embedded. And then you can think of a compact disk. And you can write with a laser 0s and 1s. And you can store this kind of megabyte of information, theoretically. So one compact disk will serve your grandparents and your grandchildren. And for 240 years, you can play music. That is the potential of molecular information storage. And Fraser was alluding to that also. But we have not realized this, because the problem is you have to address each 0 and each 1, each molecule, and they are so tiny. We can address single molecules, of course, but then we have to put a molecule here and put a molecule outside this building on the nanoscale, and then it works. So this problem has not been solved yet, although we nicely can switch. We can use similar kind of molecules to do information storage and make molecular electronics, and Fraser told about this in his designs. So this is ongoing for already more than 10 years, So once again, let's go back to the cell. And so when we look at the molecules in the cell, you realize that the molecules work together in a kind of dynamic system. So in fact, we are engaged in dynamic molecular systems, and the system is out of equilibrium. Realize when your body would be in equilibrium, we all would be dead. We are pushed out of equilibrium, of course. That's the biological machinery. And so we have to learn how to assemble. There are no engineers in your body that assemble. It's all done by instructions in the molecules and self-assembly. Trigger and response functions and motion. And when we learn how to do that, we might have responsive materials in the future, or smart drugs, or the soft robotics that people dream of, or tiny micro machines. And I will tell you about some examples how you can introduce these dynamic functions. So let me start with self-assembly. And you all know what a soap is, a detergent. When you make these amphiphilic molecules, and we designed very special ones, they can form bilayers like membranes in your cell. And we were intrigued by the idea to make very stable systems that self-assemble and to make these capsules and these bilayers. And then we build in these similar types of molecules that we used earlier in our switches that I mentioned. So they are photochemical and electrochemically responsive. And when you put these molecules now in water, you find these nanotubes. And here you see a nanotube with a tiny vesicle, a mini cell attached to it. And they are extremely well defined because the dimensions is 28 nanometers, all perfect 28 nanometers with walls of 3 nanometers. And you have these vesicles attached. And this is probably the smallest reaction tube that you have ever seen in your life. We use them to encapsulate materials, for instance a drug, and use them there for delivery. But due to the fact that we built in this core here with a responsive unit, now you see here a single tube. Here's another one. You can hit it with light and it responds to the light signal. And it can bend, and you see here the deformation and the bending, the movement of a single nanotube. And of course we can open it then, and material can flow out. So it's a stepping stone to make a nano injection needle or something that can deliver a drug in the future. This is still irreversible, what I show you here, but in the meanwhile, we have now made systems that are responsive, that can make tubes, and then reconfigure into vesicles, and go from vesicles to nanotubes again, and grow through the whole cycle. So, let me show you another example where these responsive functions were used. And this is the idea to make a nanopore or a nanochannel. And we were intrigued by the idea that you can open and close at a nanoscale and material might flow out. And for that, we went to the mechanosensitive channel of the E. coli bacteria from your body. And together with our colleagues in biochemistry, we studied this system with the purpose to make a 3 to 4 nanometer channel that can open and close with a light signal, just by switching on the lamp. And why does the bacteria use this? Normally, it's tightly closed, like a diaphragm in a camera. But it opens when the pressure builds up in the cell, and it prevents the cell from exploding. So we thought, let's engineer the system. So we took the natural system, modified at the level of the genome, built in cysteines at this specific position in the protein complex. And that is great for a synthetic chemist, because when you have cysteine units, you can hook up almost anything. So what we did is we connected these light switches, and these are spiral pyrands. They can open and close with light of different wavelengths. We introduce a strong dipole when they are in this open state, and they attract water, and most probably water flows in this pore and opens it. And so when we then put this whole engineered complex into the wall, the membrane of an artificial cell, in this case a giant vesicle, you can load it up with a dye or a drug and then switch on the lamp. The pore opens and material flows out. And you can close and open it. And all due to the fact that we build in these tiny 1 nanometer or smaller even responsive units. Respond to light. So my third example goes to smart pharmaceuticals. And the whole idea is that when you take a drug, it will be an active drug. Yeah? And it does a job at a specific position, but also elsewhere maybe. So the idea is, if we have a drug that we can switch on and off with a light or with another signal, we might have medicine with high precision in its therapeutic action. And so building it on-off switches, we thought, what are serious problems that we could face? And so the World Health Organization helps us a little bit, because bacterial resistance is called one of humanity's ticking time bombs, and almost daily you read about it in the newspapers. Or cancer treatment, all the nasty side effects of chemotherapy. So we started our journey by building in light switches in a well-known broad-spectrum antibiotic, Cipro, that is used in the clinic. And you can switch then between 2 states, and you can switch the antibiotic on and off. You can even pattern the growth of bacteria by using a mask technique due to these responsive light switches. Now, the idea is, of course, that you have an infection, you take this antibiotic, it doesn't harm your normal bacteria in your body, but only when you switch on the lamp here or a laser, you switch on the antibiotic, it does its job. We can precisely, by synthetic means, tune a half-life. So after 6, 8, or 12 hours, it switches off automatically. And when it gets into the environment, there's no antibiotic activity and no resistance build-up. This is a bit of the dream, eh? It works in the laboratory. It's still far to the clinic. And everybody who is in the field of drug design knows that it takes a long time. But maybe it's not so important for designing a drug, but more to interfere with these complex biological pathways where you can up and down regulate with a light signal. And we did the same with chemotherapy. So we took this bortezomib, which is used in the clinic. We took away this heterocycle, installed a light switch, and now we can switch on and off the proteasome inhibition, and we get some very, very strong effects. So again, the idea is that you have these tiny tumors, and you switch on the spot your activity. Will it work in the clinic ultimately? We don't know. But it is a kind of perspective that we trigger and activate these drugs, these small molecules, by building in these responsive functions and have then high spatial temporal precision in action, or in other words, making smart drugs. There's still a long way to go, in particular with respect to the light, because light that we used originally, like UV light, is a bit dangerous, and it has not a high penetration depth. In the meantime, we have made near-infrared, we and other colleagues around the world, they're making them switching with near-infrared light, and you have high penetration depth. And of course, my dream is that ultimately, with these new imaging techniques, like PET and MRI and all these fluorescent techniques, that you detect these tiny tumors, the information is then fed to the laser, and the laser then activates on the spot your chemotherapeutic agent or another drug. But this is still the future. So let me move to molecular motors. Going from switches to motors, switching back and forth, or go to a rotary motion, what I'm going to talk about, it started a long time ago. And when I was a PhD student in 1977, on page 61 of my thesis, you will find these molecules, which are these overcrowded alkenes that I showed you before, that were the basis of our molecular switches for this information storage system and the story that came later, and of course also the basis for our rotary motors. We published our first chiroptical molecular switch in JACS in 1991. And there, what we proved is that we could do 0-1 switching, where we switch from left to right or right to left. So there was directional motion from left to right or right to left in these molecules. That was the starting point for a large variety of switches, chiroptical switches in particular, and changing material properties. And then for the unidirectional molecular motor in 1991, the directional motor, the motors on surfaces, the movement of micro objects, the nano car, and here my way to Stockholm in 2016, where I brought this car. And you might wonder, we built this nano car, and I'll come to that in a second, but this is the first electric car ever built in the world, as far as I know. It was built by Professor Sibrander Strating at the University of Groningen in 1835, and I gave a replica to the Nobel Museum, together with a tiny tube of my nanomotors, but that were 1 billion times 1 billion identical nano machines. So, we all know what a motor is, an electric motor or the ATPase. And I would argue that ATPase is the most important motor in our world, because it fuels your body. It makes all the ATP in your body. And when it rotates, it's a rotary motor. It's a fantastic machine. You realize that when it does a job, it can do two things. It can either produce ATP, and it produces a lot every day in your body. But it can also act as a proton pump to pump protons through the membrane when it consumes ATP. But realize it rotates, and it rotates in one direction. And that brings me to the fundamental scientific questions at the basis of our molecular motors. And that is how to control rotary motion in the nano world and how to control left over right. Because that is crucial. If you cannot control clockwise over counterclockwise, you cannot build a rotary motor. So this brings me to another very fundamental scientific question, and that is stereochemistry. And the first Nobel laureate in chemistry was Jacobus van 't Hoff. And he didn't get the Nobel Prize for his stereochemistry, but we all learned from him and Lebel in France that molecules can have 3 dimensions and that you can have mirror images. We call them enantiomers. And then my PhD supervisor, Hans Winberg, I think he was a master in stereochemistry, and I loaned a lot to him. So why is stereochemistry so important? The essential molecules in your body, like the amino acids, the sugars, the DNA, they have all a single-handedness. Life is based on chiral molecules. That's crucial. And Albert Escher called it a signature of life. So how can you distinguish between left and right? Because that was the fundamental question we were facing. And this goes back to my childhood. When I was a small boy in the farm, we had these wooden shoes. And we all make sometimes the mistake that we step in the wrong shoe. And we sometimes have the problem with left and right. But if, as a small boy, you step with the right foot in the left wooden shoe, it hurts so much that for the rest of your life you know what the distinction is between left and right. This problem kept me busy the rest of my scientific career. And so we had to break the symmetry. And what we did is, to build these rotary motors, we built one single enantiomer and put in the proper stereochemical elements to make it moving in one direction. And you recognize here this tiny molecular machine, which was the first molecular motor, which is powered by light. It has two halves which rotate with respect to each other in 4 stages. And you see the different colors that reflect the 4 stages of a full 360-degree rotary cycle. You see here the carbon-carbon double bond. And you might again wonder, how is it possible that it can rotate? But it can only rotate, of course, like in your eye, like with the switches that I showed you before. When you hit it with light, you break temporarily the bond, picoseconds, very short period, it reforms, but in the meantime it has switched around this bond. So why do we call it a motor? First, because we have controlled motion, we consume energy, and we have control over directionality, and it's a repetitive process. Otherwise it would be a switch. Think of your car. If there's no fuel in your car, you wouldn't get anywhere. And if there's equal probability going forward and backward, you wouldn't get anywhere either. So we have to control directionality, and this is what we did, but due to the stereochemical elements, both helicity and these methyl substituents, which are stereo centers, and that control and direct if it goes clockwise or counterclockwise. So this one goes clockwise, and this one goes counterclockwise. So it's a 4-step motor. As you can see here in this animation, the fast process is the photochemical process, and the slow process is the thermal process. So we discovered it also a bit by accident, because we were working on these switches, and we were doing a quarter turn. That's normal. And then it switches back, as I showed you before. But we found an example where we didn't realize originally that it had done 2 of these turns, 2 of these quarter turns. And this is serendipity. But once we figured out that it had done a 100-degree rotary cycle in one direction, we thought, hey, maybe we can repeat it twice. And then we have a 360-degree rotary cycle. So it's a double bond isomerization, an extremely fast process by light, a slow helix inversion, a fast double bond isomerization is a slow helix inversion. 4 steps add up to a 360-degree cycle. And you might wonder, how is it possible that it doesn't go back? And that is because when you hit it with light, it goes to a metastable state or a less stable state, and the molecule wants to move on in the same direction because this is an energetically downhill process. So you pump it with energy, then it goes down, you pump it with energy, and then it goes to a 360-degree cycle. And it can be repeated and repeated and repeated as long as you have the light on. Now, what can you do with such a motor? So first, we took advantage of this cycle and said, look, maybe we can put some function there. And as I work also on catalysis, what we did is we put some catalytic functions there. So on both sides, we put a group that can work as a catalytic moiety to do a chemical reaction. And when these groups are far apart, A and B, they don't operate very effectively. So we have a catalyst, but what we make is a racemic product, 50-50 of both molecules of the product of this chemical reaction. But then, when we go through the rotary cycle, step 1, A and B come together the two groups here cooperate and we get a very effective catalyst. But because we have a helical structure, one enantiomer, one chirality, we generate suddenly a preferential one enantiomer of a chiral product. And then we can move on and we generate another enantiomer of a product. And then we go back to the original state and start to make racemate again. So here you see a function where we take the motor, we go through a cycle, and we make 3 different products with 3 distinctive stereochemistries. I'm not aware of any enzyme or natural system that can do that. And it's all due to the fact that we can have this responsive function built in. So this is our stepping stone to multitasking catalyst or doing different things in sequence, like an automated sequence of events in a robotic line, for instance. So this original motor was not much of a motor because it rotated once an hour. And you cannot do much with that. But then we made 50 or 60 different motor designs, and we could enhance the speed from 1 rotation per hour to 10 million rotations per second. So we can do everything in between, minutes, seconds, microseconds. But this is not entirely realistic, because you will realize it also depends upon the amount of energy you can get in, or how you, in what environment, are you on a surface, are you in a polymer matrix, etc. That all makes a difference, of course. So in reality, we are now at 3,000 to 4,000 cycles per second in our fastest motors. So we can put these motors on surfaces, we can move micro objects and droplets, we can have macromolecules, polymers that can change their states, and we can have single molecule movement. And I will show you in the last part of my talk a few examples how we use them. So here what you see is a surface. When you take away the glass plate from your smartphone, you get liquid crystal material, the soft material, that you see also behind the screen of your television or your PC. This is the soft liquid crystal material. And you see these waves here, like waves on the sea. This soft material, we put in a tiny amount, just a very tiny amount of this 1 nanometer size nanomotor. And on top of it, of this soft material, we put a glass rod, which is 10,000 times the size. And now, you don't see the motor, of course, because they are too small. But when you hit it with light, you see this object rotating. It rotates clockwise because the whole motion is amplified through the liquid crystal matrix and rotates this micro object in a clockwise sense. But you see also the surface changing, the architecture of this soft surface, the color changes. So this is a way to make responsive surfaces and responsive materials. We can go a step further, and this took us a long time, 5 years. We struggled with this problem, to build these motors on surfaces. So we had to put legs, stator, axle, and the rotor, and have them all spinning in concert on the surface. So we used the techniques of self-assembly to put these motors, for instance, on gold, and then we can have a kind of a windmill park where all these tiny motors rotate in the same direction. And we made them on various surfaces. And of course, this is also, the purpose here is twofold. First, to interconnect these machines, these motors, to the outside world, to surfaces, to devices. And second, again, to make responsive surfaces, because you can now move water droplets, change the shape of water droplets, etc. And of course, we got inspiration from our ancestors in our country who built 500 years ago these beautiful windmills. We did the same, but just 1 billion times smaller. But we still have to learn, because our ancestors in Holland were very clever, because they could pump the water out of our polders to keep our feet dry. We still have to learn how to use them, to put them on the dikes, and to make kind of tiny nano valves, using them as pumps. We also built this nano car, and this got a bit of attraction. The whole idea came from the fact that my students were a little bit jealous of the engineers that every year can go to Australia with their solar-powered car. And so we said, okay, let's then try. We are not engineers, but let's try to build a nano car. And you see here the frame. This is a 4-wheel drive car, 4 of these motors, and it should move over the surface. And why did we build that? This is the molecule. It was a lengthy total synthesis. That was the hard part. In total, it took us 7 years to prove actually that it could move over a surface. And of course, you might wonder, why building a nano car? But the fundamental scientific question, of course, was, can you convert rotary motion into translational motion? Can we have a rotary motor? Can we now move something along a surface? And then, can we go to single molecule motion instead of a collection of molecules? That were the scientific questions that we had to answer. There's one small point that I want to add, and that is the following. We had to control the stereochemistry of the wheels. And I don't know if you have ever realized, when you sit in your car, when you look at the car moving, the front wheels, when the car moves forward, both go clockwise. But when you are behind the steering wheel, one goes clockwise and the other goes counterclockwise. If they would have the same chirality, the car would do this. But if they have a pseudo-chirality, clockwise, counterclockwise, the car goes straight. So we built all these designs, and then we would be able to move the molecule on the surface. So when we excited it with the scanning tunneling probe tip, you can move this car over the surface in a more or less linear fashion. Now, I want to make one more point, and that is, when we think about cars, we have this idea of cars on the road. But in your muscles, all these motors, they step over the filament, as Jean-Pierre already mentioned. So more likely, the movement of a car in the nano world, is like this. So a stepping movement over a surface. This is probably a realistic picture. So you might wonder, with all these light-driven systems, could you ever propel something in your body? And I got this question often. And over the years, we have designed several catalytic systems, because the motors in your body, ATPase and so on, are all catalytic functions. So we designed chemical catalysts with the idea to propel a kind of a submarine using catalyst. And we thought, what will be the fuel? And there is plenty of sugar in your body. So we thought, let's make something that can be fueled by sugar. So what we did is, in brief, we built the nanotubes, we attached catalyst to it, and then you can see here in real time these tiny spiders moving. And you see these black dots here, that is the oxygen coming off. So what happens is these catalysts convert sugar and generate oxygen and have a kind of a rocket propulsion system. And they move, as you can see, these are tiny spiders of these nanotubes because they form tiny aggregates. And they move completely autonomous as long as there is sugar. If the sugar depletes, it stops. When you add new sugar, it moves again. I'm not touching it. This is in water under the microscope. So, will we go into this fantastic voyage where we have these tiny submarines that go through your body? Will, in the future, we have these micro and nano robots or delivery systems? Will some of the surgeons of the future be injected in your blood vein, being micro robots that go to a cell or do repair a defect or deliver a drug? I don't know. 50 years from now, it's once you know how to move, maybe. So it looks science or science fiction. But we are scientists, so I like this quote a lot. The best way to predict the future is to invent it. And I think this is what we try to do together, and also with my colleagues in this community. So let me finish by summarizing the art of building small. By programming molecules, we go to responsive and adaptive functions, trying to mimic some of the functions of Mother Nature, but not being limited by the molecules of Mother Nature, because we have endless possibilities. And then we can have induce motion and generate real dynamic systems. And I showed you here how we had a journey from switches to motors and rotary motors and propulsion systems. So this will give us information systems, as was alluded to today, responsive materials, smart surfaces, self-healing materials. So when you have a scratch in your hand or your finger, it repairs itself normally when you're healthy. So in not so distant future, you have these materials, these coatings on your car, I would predict, that once there is a scratch, the vacuoles that you can embed there, the tiny vacuoles, for instance, you can hit it with light, it opens up and it repairs itself. Or you can have coatings on your wall. Imagine, soon we will have Christmas dinners. But sometimes we have children's parties. And the coatings on your walls might well adapt in the future to the mood that you are in, or you can switch them. Adaptive catalyst, precision therapeutics, nano-scale energy converters, soft robotics. There are many opportunities once you think of the enormous possibilities when you can control dynamic functions. So with that, I want to finish to acknowledge, of course, the people that the students, the co-workers, the staff in our laboratories. And I show you this picture of the last year, this year, of my group, which is a multinational team coming with different backgrounds from physics and biology, many of them chemists, of course, from many different countries. And I can show now 10 slides with names and pictures, but I refrain from doing that because they reflect several generations of students that have passed through my group, several generations of students and co-workers that made this all possible. The colleagues and staff, the collaborators all over the world in this community, of course, the supramolecular synthetic chemistry and molecular machine community, which I own a lot to, without mentioning all the names, but I'm sure I'm going to forget a lot. And of course, my family over the years for all the support. The granting agencies, because without grants we cannot do fundamental science. And let me emphasize the Dutch Science Foundation, and again emphasize the European Research Council, who generously supports us with funds for fundamental science, which is so crucial for this kind of work, where you try to lay the foundation for the technologies of 30, 40, 50 years from now. But let me finish with one slide. And going back in time a little bit further. And that is to one of my heroes, Leonardo da Vinci. Where nature finishes producing its own species, man begins with the help of nature to create an infinity of species. And for the young students in the audience, imagine the unimaginable. I thank you for your attention.
Thank you so much, Ben, for this really dynamic and enlightening talk. I think it's already been a fantastic voyage, but we have more to come, that's for sure. And I also think that we have a lot to thank Dutch traditions for, especially maybe wooden shoes and windmills. So thank you so much, Ben. So, may I now ask Sir Fraser and also Jean-Pierre Sauvage to come back up to the stage. So giving us the opportunity to thank all three laureates and give them our warmest congratulations.